In the final step of protein synthesis, we work through translation. Now, remember where we came from. So we started this process with transcription, where we took DNA, which carried the instructions for which protein to make, and we used it to make RNA, which remember just happened to be small enough it could leave the nucleus, specifically messenger RNA. And then we just talked about the fact that the messenger RNA is then going to be used to make the protein through the process known as translation. Now, a couple of quick things to note about translation. First off, it's going to occur in the cytoplasm. So that messenger RNA is going to leave the nucleus, it's going to go out into the cytoplasm where we find ribosomes, and that's where the proteins will be made. Now, in order for translation to occur, there are several things that you have to have. First off, you've got to have the messenger, which was just made in transcription. Remember, it's important because it carries the instructions. You have to have ribosomes because remember they're the protein factories. But then you also need transfer RNA, which is a different type of RNA we'll look at in just a second. You need amino acids because they're the building blocks of proteins. And then of course you're going to need energy to power this whole thing. So we just said in addition to the messenger RNA you're going to need ribosomes. Why? Because remember ribosomes are protein factories. Now on most images of a cell ribosomes just look like little dots, but when you look at them in a little more detail you'll notice that every ribosome is composed of two subunits, a large one and a small one. Now these are formed by a third type of RNA known as ribosomal RNA, which is what makes these. But when you take these two subunits and you put them together, the messenger RNA can actually thread between the two. So that messenger that's carrying the instructions would actually fit in between these two subunits in order for the protein to be produced. Now a third requirement we said was you had to have transfer RNA. Well, what does it mean to transfer? Well, to transfer means to carry or to move, and so the question is, what are they moving? Well, in my mind, transfer RNA are kind of like Lowe's delivery trucks. You know, if I want to build a house, of course I'm going to need instructions, I'm going to need blueprints on how to build it, but I'm also going to need materials. I'm going to need bricks and nails and wood, right? So just as the Lowe's delivery truck brings the materials I need to build my house to my land, the transfer RNA brings the building blocks of proteins, which are amino acids, to the location where we're actually going to build those proteins, which is to the ribosome. Now, each tRNA has a specific amino acid to transport, so it only carries a certain amino acid to the correct location. So how do we ensure that these tRNAs that are carrying certain amino acids, how do we ensure that they put these amino acids together in the correct order? Well, the answer is actually located at the very bottom of the tRNA. Now, you'll recall that the messenger RNA that was just made from DNA, it's read 3 bases at a time. We call that a codon. Now, one of the things that you need to know is that every codon has a complementary anticodon. Now, complementary would just mean that the base is pairs. So, if you have a U, the anticodon will possess an A. If you have a G, anticodon will possess a C. Now, why are anticodons important? Well, first off, they're located at the very bottom of a tRNA. And so what happens is, if I read that the messenger contains this codon, then I know I'm looking for this anticodon, which ensures that it brings the correct amino acid at the top. So these anticodons help ensure the proper sequencing of amino acids so that I can put proteins in the correct structure so that they can do the correct function. So it's kind of like baking a cake, this whole process of protein synthesis. So let's say that you decide you want to bake a cake and you're flipping through a recipe book and you pick the recipe that you want. But you can't take that book out of the library, so what you do is you take that recipe and you copy the recipe onto an index card or onto a piece of paper that you can actually leave with. This would be transcription, just like taking that DNA sequence and copying it into messenger RNA. But that card does you no good if you don't take it into the kitchen, right? Just like messenger RNA does you no good if you don't take it to a ribosome. So you're going to take your recipe card and you're going to put it on the counter in the kitchen. Just like the messenger RNA is going to go find a ribosome and bind with it. Then you need materials. So you would then go and collect the flour, the sugar, the eggs, all the things that are needed, and you bring them to that same kitchen counter. Just like the cell takes all the amino acids that are needed to build the protein and brings them in. Now, how does it bring them in? Well, it brings them in order using tRNA. Just like how do you bring these materials or ingredients in? You bring them in using measuring cups and measuring spoons, and you put them together in the correct order to build your cake. Just like you put amino acids together in the correct order to build the protein. So very simply, the protein is put together one amino acid at a time. So the ribosome attaches to the mRNA at the promoter region, which is where the beginning is. The ribosome guides the docking of tRNA anticodons to the complementary messenger RNA codons. Peptide bonds form between the amino acids that are brought in, and as the amino acids join, they get longer and longer chains, building the protein. Eventually, protein is released once it reads a stop codon. The protein folds up on itself and moves where it's needed into the cell to do whatever function is required. So again, I'm not going to play this, but I just want to kind of get you a visual. So here's our messenger RNA. Remember, messenger RNA is read 3 bases at a time. And so as we read these, excuse me, you'll see here's our ribosome. So the mRNA fits in between the large and the small subunits. And then one codon at a time is read. The correct anticodon matches to it. And again, remember, it brings with it at the top the correct amino acid. So then it would read the next codon, UUU. It would find the anticodon, AAA, and as it binds in, it brings with it the correct amino acid, and those amino acids join, binding with a peptide bond between them. And then that ribosome just continues moving down the messenger, reading the next 3, AUA, it binds with it, UAU, bringing with it the amino acid at the top, and the existing 2 amino acids bind to this new one, making a chain of 3. And so the ribosome keeps moving down and bringing in this tRNA, binding amino acids together until eventually it gets to the very end and it reads a stop codon where the amino acid chain breaks off, and then it would fold into the protein that it's needed to be. And you can see here from your textbook, bottom page 182, this entire process of putting amino acids together in the correct order.